we will present some of the work that we recently published about how these TET2 mutations impact on the neutrophil development, and at the same time the impact that these mutations cause in stem cells regarding the response to different immunological challenges. So for example, at the beginning, we will show some of our results about how this mutation impact on the neutrophil response to different immunological challenges. And in this case, as it has been also reported for macrophages, we show that neutrophils that carry this TET2 mutation produce an exacerbated amount of inflammatory cytokines. And these pre-leukemic neutrophils that we call, they also show specific phenotype. For example, they have a reduced phagocytic capacity and also show an aberrant formation of nets, which are these extracellular DNA traps that they release when become activated. And in the case of these pre-leukemic neutrophils, this net is smaller and is more compact, which means, as we prove in this publication, that is harder to be degraded by plasma nucleases. And we think that this can be related to the pathologies that are now in the last years associated with clonal hematopoiesis, such as COPD or cardiovascular diseases, because if these nets that are released by pre-leukemic neutrophils stay longer in circulation, they can lead or likely be related with a higher risk to develop these pathologies. So this is the part that we are presenting regarding the neutrophil development and the neutrophil response in the case of the two mutations, and then we will present new data, and that is we are yet trying to put together is work in progress about the impact on the at the level of the stem cells. And in this case, it is quite interesting, because if in one side the immune progeny, myeloid cells, macrophages, or neutrophils, as we were saying, when they carry this mutation, show hyperinflammatory response. In the case of HSCs at the level of stem cells, what we reported is that the mutation is associated to a refractoriness to respond to LPS or other immunological challenge. And we believe that this is at the basis of why, upon this immunological challenge, we also observe how the mutation expands over time. Because precisely the stem cells that are mutant for the TET2 show this resistance to the immune stress.